Okay, last section. This is going to be section 4, and this is going to be the effector mechanisms of immunity. So these are all the ways that all those white blood cells, all those cells of the immune system are going to keep you safe. So here we go. All right. Shown here are some different viruses, parasites, bacteria that people get. And so there'll be a name for whatever it is, and there'll be a name for the disease. Sometimes it'll be the same, like influenza virus causes influenza, or you can just call it the flu. Sometimes they'll be quite different, like SARS coronavirus 2 causes COVID-19, or variola virus causes smallpox, or Ascaris worms, which is a nematode, causes ascariasis, which is the disease. So the cause is over here. The disease is how your body responds to the cause, plus everything that the cause is actually doing. And so it is an important distinction. A lot of medicine is based on treating the disease. And to some extent, you can treat the disease without knowing exactly what the cause was. There are some treatments that are broad enough that, you essentially don't need to know for sure ever. Although I would like to know, just for peace of mind. Okay, so innate immune responses, have potentially several different effector modules, like different ways that they can cause stuff to happen. You've got things like cytotoxicity, ability to actually kill cells. So there are CD8, or you can call them killer T cells. These definitely kill cells. NK cells will also kill cells. These are natural killer cells. But they're each looking for different things. They kill in actually a fairly similar way with perforin or something like perforin, but they are looking for different things that are wrong. So infected or stressed out or, something not being normal. So there's type 1, innate, adaptive immunity. And so this is going to be driven by these cells. It's going to be called a Th1 response. That's the most common way that you'll hear it discussed in the things that we're going to be reading. And so, Th1 response is going to be driven at making some antibodies and making also some of these CD8 killer cells to go out and get rid of pathogens that are actually inside your cells. You're also going to have a lot of activation of macrophages, which are one of the cells that goes in and they're like the heavies. They just go in and destroy whatever it is that's there. They just eat and try and wreck whatever is going on to try and keep you safe. Then you got your Th2 response. This is going to be a little bit more directed toward just antibodies. We're not going to have as much of this CD8 killer T cell over there. And this tends to be directed a little bit more toward getting rid of different kinds of parasites. We're going to have eosinophils, basophils, and mast cells. Type 2 immunity, these T helper 2 cells, Th2 cells, are going to be associated with a lot of the things that we would see as inflammation or as allergy, which is just an immune reaction to something that probably wasn't going to hurt you, but you make an immune response anyway. Type 3 immunity is a neat thing. It's by these Th17 cells, which are, the Th means it's a helper T cell. Sometimes these CD8 T cells will be called TC for cytotoxic T cells, because they kill cells. And Th17 cells make a thing called interleukin 17. That's one of their messages. And they are their own part of the immune system. They're really good at cleaning up some kinds of infection. Bacteria, fungi, they're good at bringing in neutrophils. If you're trying to make a vaccine, for example, you've got to think, well, what kind of immune response do I want? And then it's kind of a crapshoot, which of these immune responses you're actually going to get. You don't necessarily get to pick which immune response the body makes, and this can be a problem. There are cases where you would definitely want a Th1 response and not a Th2 response, where one response will kill you and the other response will clear out the infection. And there's no good way to directly manipulate what the immune system is going to do, what kind of response it's going to make. And so this is a problem with immunology in general. That, if you could just control the immune system, it's incredibly powerful, but it's kind of self-regulated, and there's not a good way to regulate it from the outside. But there are a lot of smart people that are trying to do something like this. Okay, so antibodies. What's their deal? Well, you can make antibodies against all sorts of things. So, for example, if you had a bacterium that's releasing a toxin, so now a toxin is usually going to be some kind of protein that will damage cells in a certain way. Bacteria mostly use toxins to try and break down cells so that they can eat them. They're essentially like peel an orange so you can eat the delicious segments inside. And so the toxins are there for the bacterium there, and they're there to kill whatever it is it's trying to kill. The other thing bacteria do with toxins is they will fight each other with toxins. So a lot of antibiotics are actually things that bacteria use as weapons against other bacteria. And what they're fighting over is usually either space in a particularly good place, or food, the ability to eat whatever it is they've landed on. And that's one good way to make sure you get a meal, by killing off everybody else that shows up at the dinner table. Bacteria are terrible. Nature is terrible. This is just the world we live in. But antibodies can bind to those toxins, and if they're bound to an antibody, then they can't stick to a cell, and so they can't cause whatever is bad is happening. There will be bacteria that are floating around, and antibodies can stick to those, and they can actually act like that little cocktail fork or that little toothpick, so that macrophages can grab onto them and eat them. And they can also serve as a point where you can start activating what's called the complement cascade, which is another thing that you have circulating around in your bloodstream that will kind of non-specifically kill off bacteria and punch holes in them so that they can die. And not only will you punch holes in these bacteria so they die, you will also eat them. So at the end of anything that has to do with antibodies, you have some kind of macrophage usually, or one of these phagocytes. A neutrophil could do it to some extent as well. And they'll just be eating whatever has an antibody on it. It's like they love antibodies, and, it's like putting cheese on something and then just saying, yeah, I don't care what it is underneath. As long as it's got cheese on it, I'm going to eat it. That's kind of who macrophages are on a sort of personified level, if you like. Okay, how do cells let other cells know what's going on? Well, they've got major histocompatibility complex, or the MHC. Class 1 is for pretty much every cell. Every cell in the human body can make MHC class 1. And this is where you show a little bit of what you're making today. It's like a little show and tell for the immune system's benefit. Only if you show them the wrong thing, like you show them you're making a little piece of E. coli today, then the immune system's going to come in and kill that cell. So it's show and tell, but with a business end, if you like. MHC class 2 is a version of MHC, but this is only going to be made by some cells of the immune system. So it's how they show that they are going to be infected. And there are different types of immunity that class 1 and class 2 presentation lead to. But both of them are fundamentally trying to show a little piece of whatever it is that is potentially making that cell sick, so that other parts of the immune system can see it and can respond to it. All right. So, T cells, there are killer T cells, which are like effector cells. They go in and destroy infected cells. But there are also T cells that go out and try to regulate B cells. And regulate other T cells, and regulate all kinds of other cells. These are your helper T cells. They are going to be regulators. They are the closest thing we have to controllers in the immune system, but they don't all know what the other ones are doing. There's no central control. It's just like, I don't know, a little gang leader, or a little maybe platoon leader, if we're talking military analogy. It's got a couple of cells that it's running, like a little boss in your area at work. Yeah, but not the big boss, just a little tiny one that's over your area. They have lots of different ways of regulating and encouraging the right kind of behavior. They're little tiny middle managers. Maybe that's a better a better thing. So picture somebody from the office. Yeah, just middle manager type. Yeah, there you go. That's your helper T cells. Okay, and so you'll have virus-infected cells that'll put out a little bit of a distress message, hopefully, and we'll call in a T cell. The cytotoxic or killer T cell, this is the one that has a protein called CD8 on the top. I like to think of them as being goofballs that wear around one of those sort of 8-ball hats, which, there's something kind of unhinged going on in there, but you're never sure exactly what. That's just, what I think of cytotoxic T cells in general. They are, so this is a cell, this is making little viruses. So they're the viruses coming out, and they've stained them with little antibodies labeled with gold, just so you can see where the virus is, and you can see all the little dots are around here, so those are viruses. They have an antibody stuck to them. And then here's our virus-infected cell. Here's a T cell, button up against it, and the T cell is going to latch on to the virus-infected cell, and then it is going to sort of read out whatever it is that cell is making. And if it turns out it's making something bad, it is going to kill the virus-infected cell. That is just what happens. So a killer T cell, or a cytotoxic T cell, and that's the little C right over here, cytotoxic, is going to have the CD8 protein. You'll need to learn a lot of these proteins, but we'll do it bit by bit, and we'll build it up. Yeah. We don't need to have them all today, but I want you to have seen these things so that when we start talking about them in earnest, then you say, oh, yeah, I remember that thing from Chapter 1. So it's going to be looking for an MHC class 1 that is stuck to the outside of the cell. It's going to have a T cell receptor that recognizes whatever particular antigen is going on here. And CD8 is kind of a clip that holds on to the side of this and makes sure it is an MHC class 1 molecule. And then if we score a good hit with CD8 and a good hit with our T cell receptor, then the T cell is going to release these little granules, and they're going to punch holes in this cell because there's something it has recognized that's come from inside of this cell. That's the way we get MHC. And that cell needs to die for whatever reason. Yeah. Then you got your helper T cells. So these guys are going to go around, and they're going to turn on and turn off different cells in the immune system. So a Th1 T helper cell type 1 is going to have a CD4 protein instead of a CD8. The CD is cluster of differentiation. It's like an old term. Most of the proteins that have some kind of role in an immune cell are going to be called CD something in a particular number. And so then the key is going to be trying to figure out a way to remember that number. CD4 I like to think of as like a 4-star general. These guys are coordinating, commanding. They are going around and making sure everybody is doing their jobs. So they make a T cell receptor, just like before, and they have CD4, which then reach around the side of it and bind to it. And they're still recognizing this antigen. But if they recognize this antigen, and then they're telling this macrophage, hey, what you have picked up is the thing that I've seen somewhere else. This is bad. You need to go out and find this and punish it. Activate and tell others that you have activated. Same thing it'll do, it'll show this same little peptide. It'll even take it sometimes from the macrophage and go and physically show it to a B cell and say, can you make an antibody against this? Because if you can, I'll give you a special little signal that will activate you and help you to divide, to make more copies. And so the B cell will try, and it either will or won't get that second signal, that little attaboy, attagirl, there you go, you did it, signal from the T cell. So this is a T follicular helper. It's going to the B cell follicles, like where the baby B cells hang out. And they are going to try and help B cells mature and become better B cells. So these guys are like teachers, kind of the T follicular helper cells. And there's the words up there that I'm saying, in case some of these words are kind of strange and very immunology. They're not things you run into in other parts of the world. Macrophages can also get infected. And if the macrophage gets infected with something, like here's a macrophage that has little tiny pink bacteria all inside of it, then a T cell can recognize some of these proteins that they're making from the little mycobacteria inside and say, hey, you, big dumb macrophage, have eaten something bad that you shouldn't have. You are now infected, and so you need to make a response that kills off whatever it is inside of you. And so they're going to have these little granules, these little lysosomes, and they'll just merge them with whatever is going on in the cytoplasm and try to destroy it. And they will just physically degrade whatever it is that's making that cell sick, to the point where they can sometimes actually clean out an infection. So helper T cell, Th1 type help, definitely helps macrophages do their job. Okay, so they're all different responses, and sometimes we get the immune response we want, and sometimes we get the immune response we need, and sometimes we get the immune response that would totally be good in a different situation and does not help us one bit here. So with an infectious agent, if we make a good normal response, we will actually get protective immunity. We won't get sick. If we don't make enough of a response, or we make the wrong response, then we will get infected over and over and over again by this thing. A normal response to something that's not going to hurt us, though, would be called an allergy. Normal response to an organ is called organ rejection. Normal response to your own organs, that's autoimmunity, where you attack yourself and destroy yourself. Yeah, that's really bad. Normal response, like no response to a tumor, or rather, normal response to a tumor would be to recognize the tumor as being somehow wrong and punish it, kill it, destroy it. And most cancer actually gets cleaned up this way. Now, a deficient response where we do not recognize that cell as being cancer, that's what actual clinical cancer is. Everybody has some of cells in their body somewhere that are dividing to an unacceptable level right now. An example would be, do you have any of those little brown spots on your arms, those little clusters of melanocytes? Those are actually little tiny melanomas. They are melanin-producing cells that have gone a little bit haywire and should definitely not be doing that. You should not have any of those little spots on you. Those are cancer. Did you ever have a wart or a plantar's wart on the underside of your foot? Did you ever get one of those little skin tags on your eye? I have one right now that I need to kind of get rid of. Those are all little tiny cancers. And those are just some of the many, many cancers that we can get. You would go in for a pap smear looking for little blobs of cancerous cells that shouldn't be there. You would go for a prostate exam that would be feeling to try and see if there are any extra sort of hard lumps of cancer in the otherwise it should be kind of squishy prostate. And either one of those means that you've got problems, and we need to find a solution. So, vaccine in general is a way of making immunity. And the most successful vaccines are the ones that make the right kind of immunity, and immunity in a way that lasts for a long time. Vaccines work really well. And vaccines like the diphtheria, which you might have got as a DPT, diphtheria, pertussis, and tetanus, together. Diphtheria used to be a big problem from a little Corynebacterium, which is a little thing that kind of looks like E. coli. But we've pretty much shut this down due to vaccination. Thank goodness. Polio used to be really bad. When I was growing up, I had a babysitter who had polio as a young girl in, I think, must have been like the late 20s, early 30s. She'd come from Arkansas. And Arkansas was where we had the last wild cases of polio in the U.S. And so she walked with a limp because one leg was shorter than the other. But other than that, she was fine. She did a good job babysitting me, that's for sure. But polio used to be kind of everywhere. And the people that just got away with a limp were the lucky ones. It would go in and destroy a bundle of nerve cells connected to something in your body. If it connected to your lungs, then your lungs would no longer work. And so you had a lot of people that would end up in these things called an iron lung, which is physically presses down and lifts up on your lungs to try and force air in and out, because the nerves that used to do that for you are dead, were killed by polio. In some cases, polio would lead to paralysis. And in some cases, polio would lead to inflammation in the brain, which we normally have that blood-brain barrier to keep inflammation out, but yeah, when polio gets in there, it does bad things, and that can often be just straight up fatal. So yeah, very bad stuff. Um, Measles, we have also driven down with vaccination. Now, if you were to look at more recent results, it's come up again. Yeah, thanks Jenny McCarthy for that, and all the other people as well. But this is something that we can control, but which people got worried about. There was Andrew Wakefield put out a paper that said that it's going to cause autism based on a very small sample size, and it wasn't just that he was a scientist trying to trying to bring this out. He then goes on tours and tells everybody that he can find and doesn't pay attention to larger, better run studies that come after, and essentially ends up scaring a lot of people to the point where they don't then vaccinate. And so now this thing, measles, which actually does kill something like 1 in 1,000 people that get it, is kind of on the loose in some places because vaccines have gone down. SSPE is a really bad form of measles. It stands for subacute sclerosing panencephalitis, which is when the entire brain just starts getting inflamed at the same time and shuts down. This is either 100% or almost 100% fatal. It's real bad. And this is the later consequence, a thing that can happen from measles later on. And so, yeah, this is a thing that we can vaccinate against, thank goodness. And yeah, vaccines are part of the reason why the world isn't quite as terrible as maybe it once was. Although there are things that we can't vaccinate against yet, and maybe someday. So, yeah, all right. This is the end. There are nice little movies that come with the chapter, and they will be linked at the end of these chapters. So you can go to this Vimeo link, and it will actually take you to the to one of these things, and so you can watch. Some of these things are fascinating, where you can actually see the cells from the immune system in motion. And so I would say this comes free with the book that you bought or downloaded. And so, why not get the most for your, book $/download. All right. Thanks very much. This has been the end of part 4 of chapter 1.